Okay, hopefully y'all can see the screen. If not, just let me know or put it in the chat. So welcome to our first celebration of knowledge here for Unit 1 for Dr. Cotter's Bio 205 class. For this review, I'm just going to cover some general information about the exam, what's going to happen on exam day, and then kind of go over some of the things that you need to pay attention to and study. That way you can prepare for the Unit 1 exam. And just normal etiquette, if you have any questions, feel free to raise your hand or put it in the chat. Don't talk over anybody, that kind of thing. Excuse me. Okay, so in general, the exam's going to be worth 80 points. So every exam we have is going to be worth 80 points. You're going to have about 90 minutes to complete it. It's going to be a combination of multiple choice and some short answer questions. This test will be in person in our lab, DB123, and it will start at 8:30 a.m. And the exam will close at 10 a.m. So what you should expect next week is come on in during your normal time, come in a few minutes early if you want. You're going to hand me your unit 1 study guide, and then you're going to take a seat. We already have laptops. We'll have laptops on every desk, and you'll just take a laptop. You'll start logging in with your MEID. And then once Dr. Carter is ready to open the exam, she'll give you the password. You'll type in the password, you'll start the exam, and then you'll have until the end of lecture, 10 a.m. to complete the exam. Um, for the most part, I've seen people finish the exam as soon as 15 minutes in, and some people do need a majority of the time. But I'd say on average about 30 minutes to about an hour is what's needed to actually complete the exam. So 90 minutes is usually more than enough time. Uh, if you need more time or if you have special accommodations to give you more time, Dr. Cotter has those details and she'll inform you. But for the most part, most people will have about 90 minutes to complete the exam. The only thing I'll ask you to bring is make sure that you have your MEID and password. I know, like I store mine on my laptop, so I do memorize it, but if you don't memorize it or you don't remember it, just write it down on something so you can at least log in and you're not delayed taking the exam. Okay, so I spoke to the in-person students. I mentioned, 80 points total. Koch's postulates is something that we kind of forget 'cause it was 5 weeks ago, but this is going to cover about 10% of the exam. So this is could be your difference between getting an A and a B on the exam. So biggest things to remember with Koch's postulates. Again, observe the patient for signs of illness and isolate the microbe from the infected host. By this point, you should be able to identify the differences between signs and symptoms. So you might be asked a question for Koch's postulates to give two signs of illness and two symptoms. So remember that signs are things that are observable to you as the physician, the nurse, whatever it might be. You want to look for keywords such as you observe or you record, things like taking temperature, weight, that kind of thing. Those would be signs. Also saying that you observe like a wound or you observe like phlegm in the back of their throat, redness, whatever. Those are considered signs. Symptoms, the keywords you want to look for are the patient experience tells you they're experiencing this or they feel this. So feeling lightheaded, feeling dizzy, feeling sore, or weak. Those are all symptoms. So just remember that when you get asked this question and you start writing, observe the patient for signs of illness. Make sure you write, I'm sure it's going to ask you, write two signs and two symptoms. This is going to be one of your, or should be one of your short answer questions to write down all four of the steps and then give some extra details here. First, question 2 and question 4. Remember that we're going to culture and identify the microbe in a lab using different tests. Just like in your unit or chapter 4 case study, you want to select two biological or lab tests to run for your isolate or your suspected isolate. Biggest thing that I see people mess up on is they'll pick two in step 2 and they'll give me two new ones in step 4. So you got to remember that for the tests, when you're trying to prove that your patient was infected by a certain microbe, you need to repeat the same tests in steps 2 and steps 4 to make sure that the that all that information is the same. So the same signs, the same illness, the same tests were used to determine that the microbe was in fact what you are saying that it is. So again, just whatever you put on step 2 should be identical in step 4. I know some people miss that on the case study, and I just want to like reemphasize that here so you don't lose what should be easy points. And then again, step 3, infect a new host with the same microbe and observe for the same signs of illness. Most people do okay on step 3. That's not really the problem one. It's always step 2, step 4, not having the same tests. And then in step 1, they're not specific enough with their signs and symptoms. Okay, so cell parts. So when you look through your study guide, you'll see that we have a lot of cell parts to help us split up the different types of microbes, the bacteria, the fungi, the viruses. Um, you're not really going to be asked questions on like functions. Like you're not going to be asked like what's a pilus do or what's a capsule do, things like that. What you really want to pay attention to is we're going to give you questions like on the case study. So it should be about 4, maybe 5. And there's 20 questions total. So it's going to be 4 or 5 different case studies and we're going to give you basically information. So you're going to get something like you are given these lab results and you see that this microbe has a nucleus, it has a cell wall, um, and you don't observe a capsid coat, for example. Okay, so if you go to something like page 2 of your study guide, we have a nice little chart there that split up the eukaryotes, the prokaryotes, and the viruses. And if I gave you information like, okay, it has a cell wall, okay, that could be a eukaryote or a prokaryote, but I told you it has a nucleus, so it can't be a prokaryote, it must be a eukaryote, in this case, fungus. So the cell parts specifically are not definitions, more like if I give you a list of cell parts, can you identify what microbe, either a bacterial cell, a fungal cell, or a virus, those parts belong to? So just pay attention to that. Again, we have really good charts, and I'll bring it up later in this review session. But page 2, page 3, page 5, page 6, 7. Yeah, so those 5 pages all have these awesome charts that help you differentiate between the different types of bacteria, the different types of fungi, the yeast and the mold, different types of viruses, etc. So be able to take that information to help you determine which type of microbe you have for the exam. Cell part functions. So like I said, I don't remember seeing a lot of cell part functions on any of the exams. The few key ones you do want to pay attention to, the endospore, right? So remember that the endospore is made up of 3 different layers. I'm not going to mention those layers here, but you can refer to your notes for that or go through your handouts. And remember that the purpose of the endospore is to provide long-term protection for bacterial DNA in extreme or harsh conditions for the cell. Things like the flagella allows a cell to be motile or allows it to move around. That could impact how more difficult or less difficult a microbe could be. The pilus or the sex pilus. Remember that the sex pilus can be used by bacteria to transfer genetic information, primarily this plasmid, which is not, how would I say it, non-vital information. I guess that's the best way to describe it. And so this plasmid is not necessary for survival, but it does transmit information like antibiotic resistance. And so the sex pilus is required to transfer that plasmid from one bacterial cell to another, allowing that other bacterial cell to gain antibiotic immunity. And then for the most part, these other ones are pretty straightforward. Like I said, I don't expect many questions on this. If you go to if you go to page 3, you have a list of different cell parts um, with a lot of different definitions. I wouldn't say that list is necessarily important. Um, where does it go? Give me one second. Oh, I know. I'm missing it because I gave it to another student. That's why I don't have it. Um, so I believe it was page 3. Page 3 had a list of bacterial cell parts. Um, the way I have it broken down is that I had the parts broken down into pieces that are internal to a cell, that are part of the cell envelope, and that are external or appendages to the cell. So really, the only question I would expect from that is maybe a multiple choice question or something that's like a select all. Pick, 2 or 3 parts that are part of the cell envelope, or that are external to the cell or internal to the cell. But other than that, I wouldn't expect too many, if any, definition questions, maybe like 1 or 2. So necessarily a big deal. Bacterial shell shapes, you will get at least 1 or 2 questions on this. Um, you might get asked a question on identifying what flagella type you have. So that would be one question. The one that you're more likely to get is the big things that we studied in the class so far, the strepto, staphylococcus, and bacillus. Being able to be given an image and identify if something is strepto, which is a chain, staphylo, which is a cluster or a lump, and then if it's a coccus, so it's round or circular, or bacillus, which is the long type of rods. Um, I really don't expect you to have anything on the spirillum or spirochete because we primarily deal with the staphylo and strepto type of bacteria. Um, so by now you should be able to identify it. Might be given to you as like a microscope to identify the orientation and then the shape or something like that. And then this would lead into the rest of the questions in the case study to determine what kind of microbe you have. So again, I really want to highlight this portion is you gotta be able to compare and contrast different types of microbes that are covered in the pages I mentioned earlier, 2, 3, 5, 6, and 7, and be able to differentiate if it's a prokaryote, a eukaryote, a virus. So I'm talking about prokaryote, is it specifically gram-negative or gram-positive? If it's a eukaryotic cell, is it a protozoan? Is it a fungus? If it's a fungus, is it a mold or a yeast? And then the viruses that we talked about, the naked, enveloped, and the bacteriophage or complex virus. That is I think that's honestly a large chunk of your exam is being given this information and you being able to group it into the correct microbe. If you don't have that, you're not going to do well on the exam. There's some other things, like I said, the definitions, things like that. Even the microbe names. So kind of like in your case study, you're going to be giving all this information. You're going to give us the answers, and then we're going to ask you what is the microbe that is causing this illness. You're going to be given the list of potential suspects, just like you have it on the case studies. And so you shouldn't have to worry about spelling because the spelling or the microbe is already on that list. So you will lose points if you misspell it because we're giving it to you in the spelling there. Um, I don't recall if the exam allows you to italicize or underline, but if it gives you that option, italicize and underline just to be safe. Um, okay, viral replication and infection. This is one, as I was reviewing the Chapter 7 case studies. Some of y'all did struggle with this, not so much that you didn't get your steps, but the key thing about this question, and it's most likely going to be another short answer question, is that you have to be very specific when you're talking about the 6 steps. So for example, give me a sec. If you say that step 1 is attachment, and I'm telling you that you have HIV. So you have to tell me step one, attachment. HIV uses GP120 to attach to CD4 cells or helper T cells. For influenza, I would tell you, influenza uses the hemagglutinin viral spike to attach to respiratory epithelial cells. That is the verbatim words you should be using as you describe the 6 steps. Another thing that I realized that I saw was parts of like replication. So in our notes, we show that viruses can be either DNA or RNA, never both. But a lot of the replication questions I saw said this involves replicating DNA and RNA or RNA. That's too broad. If I give you these 2 viruses, and these are the only 2 viruses we're going to talk about and be tested on in this class, HIV and influenza are both RNA viruses. So the only words that should be written in step 4 are RNA. We're replicating the RNA of the virus, if that makes sense. Entry, again, HIV enters through the GP41 viral spike. Influenza enters through the neuraminidase. Technically, it's a spike, but it's technically an enzyme. But if you just put the neuraminidase viral spike, it's fine. It'll be an acceptable answer. On coding, that's the same for both. That you're removing the capsid coat. Replication. So HIV, I saw this also on the case study. HIV is one of the retroviruses. So when you're talking about HIV, you're gonna mention HIV uses reverse transcriptase to copy its DNA back into RNA, and then that RNA back into DNA, and that DNA is going to be used to make new RNA, new proteins to then make the proteins for the HIV virus. For influenza, it is a straight RNA virus, so it's just going to have RNA into proteins using RNA polymerase. If it takes you a couple practices to get this, that's okay. This is probably your first time being exposed to terms like this. But if you don't want to lose points on this question, you need the specifics. GP120, GP41, reverse transcriptase for HIV, and then hemagglutinin, neuraminidase, RNA polymerase for influenza. For assembly, don't just say that you're assembling viral proteins. What viral proteins? So remember, we're making a brand new capsid coat, and we're making new viral spikes like GP120, GP41, hemagglutinin neuraminidase, and it's also making the either reverse transcriptase or the RNA polymerase. So if you write all those down for assembly, you'll get full credit for it. And then release, both of these viruses are enveloped viruses, so they need that outer envelope. So they'll release by budding. You shouldn't really have to worry about lysis. Those are naked viruses. They kind of just keep multiplying in a cell. And when the cell gets too swollen or too big, the cell just pops and releases all the viruses at once. I have a question. Give me 1 second. Yeah, so the question is, will this potentially be a short answer question? I will tell you this is potentially a short answer question. The reason I say potentially is because Robin does move some questions into she has multiple choice and short answer questions. And so depending on how she wants to structure the exam, she might give you a specific way to do it. Honestly, what I expect for this question, just based on how people did on the case study, this will probably be one of the short answer questions. So you might be asked, maybe not be like, let me see, how would I say this? It may not be like one long question to tell you all 6 steps. It might say, hey, for step 1 of viral replication, what does this microbe use to attach? And then you're writing in GP120, GP41, or sorry, GP120. Or it could say, what are the proteins that HIV uses for viral replication? And that's where you're saying, I've got GP120, GP41, reverse transcriptase, that kind of thing. So to answer your question, it is most likely going to be short answer. It's just I'm not sure how she's going to write it in. I've seen it where it's just write down the 6 steps with the specifics, and that's like a, let's call it a 12-point question or something. I've also seen it where she breaks it apart where it's like 2 points, you write attachment, and then you'll write GP 120. And then the next question is going to say, what is step two? And you're like entry, and it uses GP 41. And then question three would be uncoding, releases the capsid code. So it might be one long question that wants you to add all 6, or it might be like just one question asking you for one step of the viral replication chain. It will count only for um, the microbe name. Oh, so sorry. The question was the spelling, will spelling count? The spelling will only count for, the only thing we care about is the microbe name. Um, the other thing that you might get dinged on is like, all right, if you misspell hemagglutinin, it's a weird spelling, you're probably fine. If instead of saying GP 120, you put GP 12, you're probably going to get that wrong because that's not correct. Um, but things like hemagglutinin, neuraminidase, transcriptase, polymerase, you're probably okay misspelling it. She'll figure that part out. But the spelling does matter when you're talking about the actual microbe name. If you have to write out Staphylococcus aureus, Escherichia coli, um, uh, Streptococcus pneumoniae, whatever it might be. Like I said, for those, you'll be given the list of potential suspects. So worst case, you'll be able to scroll down, read the microbe name, and then go up and type it up. So you shouldn't get it misspelled because we're kind of giving it to you. Does that answer your question? Okay, let me know if it answers your question. Yep, okay, perfect. Okay. And honestly, that's all I really had. Again, it's a very quick overview. Couple things to note. The Unit 1 study guide, like I mentioned, that's going to be due, handed to me on Monday. That study guide is your official exam study guide. So anything on the Unit 1 study guide can be tested on. Um, and so just review everything on it. But like I said, the most likely questions that you're going to get, I'm guaranteeing you're going to get a viral replication question. I guarantee you're going to get a question on Koch's postulates. Koch's postulates is most likely going to be a short answer because I've never seen it not be a short answer. Um, and viral replication is most likely going to be a short answer just based on how the class did on Chapter 7's viral replication case study. I expect that to be a short answer as well. Some of the other stuff might be like 1 word, 2 word answers, or it's going to be multiple choice. Um, Other than that, that's really all I had. Does anyone have any questions? Any other concerns or anything about how the tests are going to go, what time to show up, that kind of thing?
I have a question, Luis.
Yeah, what's up?
For the part about the cell parts and functions and how that one we should really learn how to identify them. Like for example, you give a part or a structure or an organelle, whatever, and then we have
hmm
to identify that. Can
hmm
we also anticipate that you might give a picture and then like point at a picture and then we have to identify what part of that cell structure is on a picture, or should we not anticipate that?
Uh, Honestly, I can't say. Again, there's always a first time for everything.
Hmm.
I can't say I have seen a picture, like if I gave you this picture, for example,
Hmm.
I have not seen that as a question at all in five
Okay.
years. So, I mean, first time for everything, but I think you're very unlikely to get something like this where it's point out the cell parts.
Okay.
You're more likely going to be given questions more related to here are the cell parts, what type of microbe does this belong to, kind of deal.
Okay.
Because ultimately, that's really what we're testing on. Like this is kind of Bio 181, 182 review. We
Hmm.
really care if, okay, we don't care that you can identify the structures. We care if you can take those structures and then start differentiating between different microbes, if that makes sense.
Yeah, that makes total sense.
Okay,
I
yeah.
have another question for you,
Yeah,
Louise.
go for it.
For the Koch's postulates,
Mhm.
I'm looking at the study guide and I do remember your many repeats of the study guide is fair game for the test.
Yeah.
But just because I can't help but ask this question, do we also have to No, like the other scientists that we talked about on first day of lecture outside of Cokes, or should we just focus on Cokes and then understanding his postulates?
Um, if I could give you, if I had to take a bet, I
Hmm.
would guarantee you you'd have zero questions on anyone outside of Coke.
Okay,
So I have, like I said, it might be like the one question, but
sure.
really we care about the Coke's postulates because that's kind of how um, we'll apply Koch's postulates as we go along and analyze things.
Okay,
The other important players in the game are very important, but that's not really relevant. And again, I have not seen a question of anyone else outside of Koch in my 5 years here.
okay, sounds good.
So,
Thank you,
yep,
Louise.
no problem.
Those are all my questions.
Awesome. Appreciate it. Anybody else have any questions? You can throw them in the chat or say them out loud. Um, okay. If you don't have any questions now, or if you think of questions later or something, just feel free to message me. Again, I'll post this for everyone else. So if you want to come back and watch this later, you're more than welcome to. Again, biggest thing for next week, just need to have your study guide so you can get your extra credit points, and bring in your MEID and or have your MEID and password so you can log into the computers efficiently. Other than that, that's all I have. I'll let you all go. Enjoy the rest of your night. Thank you for joining, and I will see you all next week.
Thanks, Louise.
Yep, thank you.